Turns out, creating life from nothing is easier than we thought. Hey, my name is Michael. I'm a 17-year-old genetic researcher at the Knowledge Society, and I'm here to talk to you about synthetic biology. Have you ever imagined if you could create life just from computer code, breaking the barrier between computer language and actual biology and living things? Well, synthetic biology takes this idea as pretty cool. Let's do it. So in synthetic biology, there are two fields that we look at. Number one is called artificial genome sequencing, or AGS. And this is basically creating entirely new genomes from our knowledge, typing them up, and then getting them into the cell. And the second area we're going to look at is something called DNA computing. So let's start with creating entirely new genes from just what we know in a computer. So you might be thinking to yourself, how the heck are we producing new forms of life without having traditional forms of life to come from? Well, this is where DNA printing comes in. So instead of having one really overpriced ink cartridge, DNA printers have 4 cartridges. And let's be honest, for what you get, you're paying the right price. So the 4 cartridges naturally are the 4 DNA base pairs. Adenine, or A, thymine, or T, cytosine, C, and guanine, G. So when the scientists are ready to import the genetic data, they import the genome sequences into the data printer, and then the DNA starts printing out DNA. Now if you think about it, this DNA is technically synthetic DNA. Humans themselves coded it up. We may have taken some inspiration from nature here and there, but it's still ours. Right? Once we have this printed DNA, we can then insert it into the cell with different means, and boom, we have a virtually hacked cell that can do what we want it to do, and ideally still be able to functionally survive, and most importantly, divide. So back in 2014, scientists produced the first ever strain of completely synthetic DNA containing cells. They manipulated E. coli to have an entirely man-made genome, which we call Cynthia, or first synthetic. So in the words of Dr. Benjamin, the scientists who created Cynthia for the first time, Cynthia is the first form of life on this planet whose parent was a computer. It had some problems. Cynthia couldn't divide that fast, and it wasn't really that healthy. So this is where optimization had to happen. By reducing the amount of extra genes that this bacteria had, scientists managed to create a more effective version of Cynthia that was healthier and could divide even faster. So scientists did 2 things to optimize these cells to make them more survivable. Number one was to use CRISPR-Cas9 to be able to edit faulty genes and make sure that the entire genome was relatively stable. And number 2, by understanding how the essential genes of a cell work, scientists deduced the genome of Cynthia 1 to a more minimalistic but effective survival kit for the cell to have in order to live healthily and still divide. This room could sure use Perfumer. And scientists in Switzerland decided to tackle this problem head on. So when companies are looking for scents for different perfume flavors, they're actually looking at specific proteins that create the scent. That are responsible for the scent. So let's take, for example, this rose perfume that I desperately want to clear up this room scent. So this nice scent of the rose perfume that we all know is responsible by one single liquid called rose oil. So how things currently work right now is that you have to take actual roses, deliver a ton of roses from Turkey, and then ship it halfway across the world to wherever you have to make the perfume. This is really inefficient and also really expensive. So synthetic biology came up with a nice solution. By taking the gene that codes for rose oil in the actual rose flower and then implanting it into an E. coli cell, scientists managed to create a bacterial population that actually produce rose oil and smell like roses. So not only does this allow a bunch of roses to have their lives spared, but it also allows perfume companies to produce their own scents and their own unique flavors in-house. So they don't have to spend time and money to ship this stuff halfway across the world. So I'm not here to just talk to you about rose oil. Cells are literally molecular factories that we can use now to our manipulation to produce what we want. Scientists are looking at how they can produce the important things like biofuels and different types of drugs that these bacteria cells, when manipulated, can produce on a mass number and then clone themselves to produce even more. And what we get is an entire culture of these molecular factories producing whatever we want them to. And that's pretty sick. So a couple of years ago, scientists actually decided to upload an MP3 file of Martin Luther King's I Have a Dream speech into DNA. And they not only did they get it in full quality, but they also got it in full length. It's a crazy proof of concept that might see DNA actually being used as the next generation of hardware to store big data. So people will be people. And although 250 million gigabytes of data sounds pretty crazy for 1 gram of DNA, they want to up the game even more. So how else are they going to make DNA more efficient at storing data? Well, What about completely reinventing DNA altogether? So a professor named Dr. Roseberg at the Bainer Institute decided to take the 4 base pairs of DNA and revolutionize DNA as we know it by adding 2 more. So the A, T, C, and G pairs that we regularly know were then added onto with an X and Y pair. So unfortunately for us, unlike A, T, C, and G, adenine, thymine, cytosine 1, which have like regular English pronounceable names, the scientists decided to add an XY and just keep the original names. So say hello to D5-NAMTP and D5-6. We're working on it. Now, these are fundamentally bonded differently to each other than the regular ATCG bonds, as in ATCG are bonded by what we call hydrogen bonds, which are relatively weak. But X and Y are bonded by a little bit less stable bonds, known as hydrophobic interactions. Now, all you have to know here is that hydrophobic interactions makes the XY basing not as ideal as ATCG, they just do what's best, but it's getting there, okay? So why are we increasing the base pairs of DNA? Well, 2 main reasons. Number 1, to increase the amount of data that DNA can store beyond that 250 million gigabytes per 1 gram. Crazy. By adding 2 new base pairs, we're increasing the amount of possible amino acids that these cells can produce in the protein synthesis process. So let's back it up. 6 base pairs, exponentially increasing the amount of data we can store in DNA, and exponentially increasing the amount of stuff that we can do with synthetic DNA and thus do with synthetic cells. It feels like synthetic biology. Good science requires good ethics and good laws to work along with it. Only when we have the benefit of humanity in mind can we make tangible steps to solving some of these big problems that impact billions of people's lives around the world.